All the king's men. Satellite gliocytes. Satellite gliocytes are glial cells in the ganglia of the PNS, both sensitive and autonomous. Satellite gliocytes are small flat cells that form a membrane around each neuron, creating a separate structural and functional unit consisting of a neuron and accompanying glial cells. So, each neuron cell body is fully enclosed by its own sheath of supportive satellite cells. In the satellite gliocytes, electron microscopy reveals a new nucleus rich in euchromatin with one nucleolus and cytoplasm with organelles. Satellite gliocytes are interconnected by gap junctions, and sensitive neurons send numerous thin processes in the invaginations of satellite gliocytes, which can facilitate the exchange of chemical signals between cells. Structural and functional units are separated by thin layers of connective tissue. Monitoring the microenvironment of neurons. It is known that there is no blood-brain barrier in the ganglia, and various substances pass both through the wall of the fenestrated capillaries and through the satellite gliocytes layer. However, the latter can slow the diffusion of large molecules and partially control the microenvironment of neurons. There is evidence that satellite gliocytes have a mechanism for uptake of gamma-aminobutyric acid, glutamate, and possibly other neurotransmitters, which also allows them to control the microenvironment of neurons. So they help to regulate and stabilize the environment around nerve cell bodies. Effect on synaptic transmission and autonomous ganglia. The difference between the sensitive and autonomous ganglia lies in the fact that the latter has not only bodies and processes of neurons, but also chemical synapses. Satellite gliocytes are in close contact with synaptic botons, which allows them to influence the synaptic transmission and processing of information in autonomous ganglia. Satellite gliocytes have receptors for ATP and therefore can respond to the release of this neurotransmitter by neurons. Like all the PNS gliocytes, satellite cells develop from neural crest cells, with boundary cap cells being an intermediate stage. Boundary cap cells form clusters on the surface of the neural tube and later on the developing spinal cord at entry and exit points of peripheral nerve roots. After the major neural crest ventrolateral migratory stream has taken place, boundary cap cell-derived cells migrate along peripheral axons and colonize spinal nerve roots, peripheral ganglia, peripheral nerves, and nerve terminals in the skin. Thus, satellite cells, Schwann cells, some terminal Schwann cells, and sensory neurons in dorsal root ganglia are derived from boundary cap cells. It was shown that in rodents 30% of parasites in the skin originate from boundary cap cells. Thank you for attention.